on recent trends in cancer research and its management. I am Sagar Kumar, a research scholar from University Department of Zoology, LNMU, to present here about likelihood of the ability to discern the taste of phenylthiocarbamide as a genetic material. First of all, we also know what is phenylthiocarbamide. It is a carbonic compound which is solid, and when we taste this PTC, this is also known as PTC, when we taste it, it tastes bitter in taste for those who have that particular gene, and it is tasteless for those who does not contain that particular gene. Next slide please. Now its chemical formula is C7H8N2NS. Its molar mass is 152.22 gram per mole, and it is available in a paper format, a paper strip. You can find it in the market. Its appearance is white to slightly yellow in color, and its solubility, when we are talking about its solubility, then it is soluble in boiling water. That means we need a high temperature for make it soluble. Next. PTC testing, a Mendelian trait. The phenylthiocarbamide testing is also called as PTC testing. It is genetically controlled ability of testing. It means the tasting capacity of PTC is controlled by a particular gene. We cannot say that if you are tasting a food, then we can taste PTC as well. The PTC tasting ability is Mendelian trait governed by a single gene having two allelic forms. The dominant T is for tasting and the recessive T is for non-tasting. The person who contains the dominant T, that is homozygous T, will be the testers, and the person who does not contain that dominant T, it means they will contain the recessive homozygous T, will be the non-testers. And that particular gene, you can look at the picture. There is a particular gene present in my tongue, which is code for a protein. That PTC, when we test that, then that PTC got bind with the protein, and we will taste it bitter or tasteless. Bitter for those who have that gene, and tasteless for those who does not have it. Next. Modulators of tasting ability. Modulators are those who alters the natural capacity. So modulators of tasting ability will alter the tasting capacity of a human. It may be some hormonal modulators such as the sex hormone, and maybe the other genetic factors such as the level of dithyrotyrosin in the saliva. Next one. Prevalence of testers and non-testers. According to R. F. Fisher, sir, he hypothesized that PTC is maintained by balancing selection, as a result of which heterozygous have a large representation in a population. It means that we have the dominant homozygous and dominant recessive homozygous. But we cannot extract that heterozygous person, because they also play a partial role in testers. So the frequency, you can look at there, the frequency of testers versus non-testers are 70.58% and 29.42% respectively. Most of the testers being heterozygous with respect to the testers, non-testers alleles. I say that in the testers it is 70.58%. In the 70.58%, the homozygous dominant and the heterozygous will take a partial role. Next. General importance of PTC testing. The ability to test or not to test has no direct correlation with the health of an individual, and PTC is not found directly in food content. I said it, that ability to taste, capacity to taste PTC is not dependent on our health or our fooding capacity or our fooding habits. It is regulated by genetic material. The frequency of testers and non-testers in a population and comparison of the same between population can give a fair idea about the population structure, such as the level of inbreeding, heterozygosity with the aid of other marker, and recent migration events, etc. Through this graph, this is the graphical presentation of frequency of non-tester alleles with the different population. You can look at here that it is very fluctuating with the number of population. So it says that the testers versus non-testers is not fixed at every places. Next. This is the graphical presentation of a percentage of non-testers. And India is at the 32.8% now. Next. Other possibilities of PTC testing that can be utilized. This phenotype has captured the interest of many because the ability to taste PTC is correlated with the ability to taste other substances, some of which are toxic, therefore testers would avoid such substances. Suppose that we are the testers of PTC. Then if we can test PTC, it is possible that we could test the other toxic substances. So if we test that toxic substances, we will particularly avoid that, and we will not take that with our food or with any else. And in that way, we could prevent ourselves from being diseasable, being ill, and we could protect ourselves from that toxic substances. Consequently, variation in PTC discernment may influence dietary pattern and can have a correlation with susceptibility to diet-related disease such as cardiovascular disease. So, a cardiovascular disease or thyroid, it is somewhere depend on our fooding habit, somewhere depend on our habits of eating or taking water, taking foods. So, through PTC testing, we could avoid that and prevent ourselves from cardiovascular disease and thyroid as well. Next. Can PTC testing be correlated with susceptibility to cancers related to the gut? So, we take food and PTC directly correlated with our tasting capacity, and tasting capacity is directly related to our fooding habit, because suppose that something very spicy, we would like to eat spicy things, and without knowing whether it is toxic or not toxic. So, and we consume that food, where it goes in our gut, and there comes the case of gut cancer. So it is mentioned here, although studies correlating PTC testing sensitivity, certain disease conditions are numerous. Fewer studies have focused on its relationship with certain types of cancer, such as stomach cancer. More because PTC testing non-testing ability directly influences dietary pattern. Dietary pattern is a strongly implicated factor in gut cancer. Therefore, PTC discernment may prove to be a useful genetic marker in preceding individual susceptibility to some types of cancer, and future studies on PTC gene, even at the molecular level, may give new insight into the strength of this gene as a possible genetic marker of cancer. I said it that if you taste PTC, then we can taste a toxic substance, and if we avoid that toxic substance, we are directly avoiding the food which can affect our stomach, which can affect our gut. and in that way we can protect our gut from cancer also. So PTC can be beneficial for gut cancer prevalence. Next, possible future directions. What will be the possible future for PTC? We can say that the PTC gene can be further mined to look for other variants alleles at the molecular level. We could do our discoveries, we could do our practicals on further on PTC, and in that way we can find another way to protect protect our surroundings from being other diseases. This can be correlated to taste preference and dietary pattern. Our dietary pattern can be maintained with the help of PTC. Further association studies may be done to understand the role of PTC in gut-related cancer. And further studies will happen if we study on PTC. We do some practicals, some laboratories, and then we can come to know that PTC will play an important role in the prevalence of gut cancer as well. Through our presentation, I just hope that PTC will be proved as a discerned susceptibility for the cancer person. Thank you. That's it from
Thank you so much, Mr. Sagar, for your valuable participation. It was indeed a knowledgeable presentation. So now we will have a short break, and I would request all our guests to kindly have some snacks or get refreshed or whatever seems convenient to them. We will resume with our other technical sessions.
His title of talk is Transcription Factors in Myeloid Differentiation and Leukemic Cancer Development. Proteomic system biology of acute myeloid leukemia reveals that leukemic fusion protein AML1ETO target a CEBP NM23 pathway. You are welcome, sir, for your talk. Just we are waiting for all the panelists. So sorry for that.